Hi everyone. So in this video, Tim and I are going to talk about the equilibrium experiment that you're going to be doing. And depending on where you are in the lecture course, what you're going to find out is that the equilibrium is a very large part of general chemistry too. So here is an opportunity for you to explore concepts of equilibrium experimentally using the iron thiocyanate reaction. This reaction is done everywhere, not everywhere, but at lots of different general chemistry labs. So let's talk generally about what an equilibrium reaction is. So let's say that we have small a, A's, aqueous, plus small b, B, aqueous, and it's in equilibrium with small c, C, aqueous, and small d, D, aqueous. So this is an equilibrium reaction where the reactants are forming products, but then the products are also reforming reactants. Now what's important to remember is that at equilibrium, what is equal is not the amount of A and B and C and D. What is equal is the rate of A and B forming C and D, and the rate of C and D forming A and B. So since their two rates are equal, we can write what is called the equilibrium constant expression. And that is K EQ equals products over reactants. So it's the concentration of C to the little c, concentration of D to the little d, over concentration of A to the little a, concentration of B to the little b. And this equation is derived for you in the lab manual and in the textbook, but essentially it's based on the fact that the rate of the forward reaction and the rate of the reverse reaction are equal. So what we're going to actually investigate in this experiment is the type of reaction that is occurring. Now, up to this point in general chemistry, you've probably taken for granted that the reaction that's on the paper is the reaction that's occurring. But oftentimes in chemistry, we don't actually know the reaction that is occurring. And one experimental way that we can do that is by testing out an experiment and seeing which reaction is occurring by looking at the rate constant. So let me explain. So our choices here for the two reactions that could be occurring are, we have Fe3 plus aqueous plus SCN minus aqueous is in equilibrium with FeSCN 2 plus aqueous. And we call this, I believe, pathway 1. So this may be the reaction that's occurring. Alternatively, if we call it pathway 2, what could be occurring is we have Fe3 plus aqueous plus 2 SCN minus aqueous is in equilibrium with FeSCN 2 with a positive charge, aqueous. So maybe pathway 2 is the reaction that's occurring. Well, if you remember back to the equilibrium equation that we wrote a minute ago, we could write the equilibrium expression for both of these reactions. So for the top one, the pathway 1, KEQ is equal to the concentration of FeSCN 2 plus over the concentration of Fe3 plus times the concentration of SCN minus. And here you'll notice that all the stoichiometric coefficients are 1, so these are all raised to the 1, or said another way, there's no number up there. In the bottom case, the equilibrium constant expression is going to be different. Here we have KEQ is equal to the concentration of Fe parentheses SCN 2 with a positive charge over the concentration of Fe3 plus times the concentration of SCN minus squared, because there's two of them. So here we have a different equilibrium constant expression. So that's great. They have different equilibrium constant expressions, but how are we going to use this information in order to determine which pathway this reaction is undergoing? Well, how we're going to do that is we're going to measure these reactions at several different SCN minus concentrations. In fact, we're going to use 5 different SCN minus concentrations. And when we use the 5 different SCN minus concentrations, we can then calculate the equilibrium constant for 5 different trials. We can do that for pathway 1 and for pathway 2, and we could see which of the 2 pathways has a constant equilibrium constant. So the equilibrium constant should be the same independent of the SCN minus concentration. So as we vary the SCN minus concentration, the KEQ should not vary if it's the correct reaction pathway. Now you may remember that what we do statistically to calculate precision or reproducibility is find RSD. So what we're going to do is we're going to calculate the KEQ 5 times for this pathway and 5 times for this pathway. Believe it or not, you can use the same experimental information, just do different math. And then we're going to calculate the RSD. The one with the lower RSD is the pathway that's occurring. So next what we're going to do is we're going to look at how to do this. And it turns out that the reason that so many people are using iron thiocyanate is because iron thiocyanate is kind of a rusty brownish reddish color, and it is able to be it's possible to measure its absorbance. So therefore, we can use visible spectroscopy to monitor the concentration of the iron thiocyanate product. Then, knowing the initial concentrations of Fe and SCN-, we can use an ice table in order to determine all of these equilibrium concentrations and calculate the KEQ. So the next thing that we want to do is we want to look at how we're going to actually determine the iron thiocyanate concentration. So what may not be surprising at this point is that what we're going to do in order to measure the concentration of thiocyanate in solution, iron thiocyanate, whether it's FeSCN2+ or the other one, is basically we're going to use a calibration curve. Exactly like you did for the red food coloring, except for where you have a different product here. In order to do that, your TA is going to make you 0.5 times 10 to the minus 4 molar, 1 times 10 to the minus 4, and 1.5 times 10 to the minus 4 molar solutions. These will be provided for you. The TA will make 100 milliliters of each of these solutions. The way that these solutions are made is we use a huge excess of iron nitrate. This, by Le Chatelier's principle, drives the reaction to completion. So therefore, we can actually know the concentration of the iron thiocyanate product. You will then, the TA will make about 100 milliliters of each of these solutions for the entire class. You will then fill vials with each of these solutions and measure their absorbance. You will then plot concentration on the x-axis versus absorbance on the y-axis and generate a calibration curve. This calibration curve will then be used to measure or to determine the concentration of iron thiocyanate product in a series of solutions that you'll make. So now I'm going to turn it over to Tim, who's going to talk a little bit about how you're practically going to make your absorbance measurements and then the solutions that you're going to make in order to investigate the equilibrium constant at several different concentrations.
As Colin said, I'm going to go through some of the practical parts of the lab, including a refresher on how to make the measurements correctly, and show you what to do with the microlab so that you can make your measurements. So first we need to open the microlab software, because that's what we'll be using to make our absorbance measurements, as always in this course. Once our microlab software has opened, we're going to go through a series of steps that may be familiar to you. So the first thing we need to do is select spectrophotometer, and you can either double click it or click OK. And then we need to force the microlab to do what we call the long blank, if you'll remember from the past. And to do that, we need to click on the Select Wavelengths button, then Clear All, and choose whatever wavelength seems good to you. I tend to choose the 502, but it doesn't actually matter which one you choose. And then we'll click OK a couple of times and hit Read Blank. Now once we hit Read Blank, the blank will go relatively quickly because it's just checking the one measurement, and this is the blank of the 502 of my vial of water that I have in my microlab right now. So once we do that, we click on the little X to close out that experiment, and then we hit File New, and we double click on spectrophotometer again. This time we click select wavelengths, we make sure all of the wavelengths are selected, and we click OK and OK again. Now when we hit read blank, we will see the microlab do the blank for each of the wavelengths of lights that it has, and we'll see each of the columns in the window here. We'll see them rise and fall as the microlab calibrates itself for the blank water sample that I have in the microlab. Please remember that it's really important that you see this longer blank take place. It should take a couple of minutes in order for your blank to take place. If you click read blank and your blank happens instantly when you have all the wavelengths selected, or nearly instantly, I mean less than 10 seconds or so, you'll know that the microlab is not doing the correct blank and your experiment will not work. So it's really important that you go through all these steps of selecting just the one wavelength, reading the blank at that one wavelength, closing the experiment, opening a new experiment, making sure all the wavelengths are selected, and running your blank a second time. The simple reason is if you don't do that, the experiment simply won't work as it's supposed to. So we see here that the microlab is reading all of its LEDs to get the blank for each of them. If a couple of your peaks are a little lower than the others, like our 555 peak is relative to the peaks next to it, that's okay. Don't worry about that. That's just a function of how strong the LEDs that the microlab uses to do its readings. So now we're reading our last wavelength, and we'll see our progress bar on the left-hand side of the screen will fill up, and once it fills once, it should empty and fill a second time, and once it fills the second time, our blank has been completed. So there it is. So we have our blank. Now, since we're going to be working with absorbance, we do of course want to click on absorbance so that that's the measurement that we're looking at for each of our readings. So now I'm going to go to my microlab and I'm going to take out my blank water sample and I'm going to put in a sample very similar to one of the ones you'll use in the experiment when you perform it, in order to show you what the spectrum looks like of the mixture of iron nitrate and potassium thiocyanate. So I'm going to click add, and I'm going to name this example because it's just me showing you an example, and you can choose to enter a concentration or volume, but you don't need to. I would recommend using your sample ID in order to keep track of which sample is which. So when I click that, it'll say reading data, the bar will fill up, and I'll get my spectrum. So now I have my spectrum for my my solution, and I can use this to get the data I need in order to perform the rest of the experiment. So to get the correct data, if we look over here, the absorbance says that it's 0.14, and that is not the number we want to write down. What we want to do is we want to click on the lambda max, in this case 465 because it's the tallest peak, and when we do that, the absorbance number will change to be the absorbance of that particular peak. So in this case, my absorbance is about 0.6466, which if we look at our graph, our axis on the y-axis, that seems about right to me. So that's how you'll do each of your samples. I'm not going to spend more time on that because at this point you're probably relatively familiar with performing the spectroscopy. So before I turn it back over to Colin to talk about the beginning of the math, I just want to go over with you the solutions you're going to make in the lab. So when you perform this experiment, there are going to be 5 solutions that you're going to make. These solutions will all need to be run in the same vial for the same reason that you've needed to use the same vial in the past, and that's that different vials will reflect or refract light differently and cause different measurements to be obtained of the same solution. So we need to make sure that we're using the same vial for each of these 5 solutions, and in the first solution we'll take, or in each of the solutions rather, we'll take 5 milliliters of the 2 times 10 to the minus third molar iron nitrate, which can be found in the lab, and to that we will add a different amount of 2 times 10 to the minus third molar potassium thiocyanate. And each time, once we've added our 5 milliliters of iron nitrate and our varying values of potassium thiocyanate, we will add enough water to bring the total volume of the solution to 10 milliliters. And the reason for that is simple. As Colin said in the first part of this video, we're concerned about concentrations. So if we're concerned about concentrations, the only way to keep one of our concentrations static throughout the experiment is to keep our total volume static. So if we keep our iron nitrate at 5 milliliters the entire time, and we keep keep our total volume at 10 milliliters the entire time, we know that the concentration of iron nitrate at the beginning of each of these experiments is going to be exactly 1 times 10 to the minus third molar. And when it comes to the potassium thiocyanate, the concentration will change as we perform the different experiments because we use a different amount of potassium thiocyanate while keeping the total volume constant. And we'll talk more about how to calculate those numbers in a few minutes. First, I'm going to turn it back over to Colin, who will discuss with you some of the math that you're going to need to do before we go back to Excel and look at how we're going to specifically calculate that for this experiment.
So now that we have made each of the solutions, we have to figure out how to find the equilibrium constant. And it turns out that finding the equilibrium constant is a challenging thing to do. And hopefully you've already seen this in the lecture course, but if not, we want to go through how to find the actual equilibrium constant. And this involves using an ice table. So what we're investigating here, I'm going to do this for one example of pathway 1, is that we have 1 iron 3 plus 1 thiocyanate yields FeSCN2 plus aqueous. So this is our possible pathway. And as we talked about before, this is the equilibrium constant expression. But the problem is, one problem is, we need the equilibrium concentrations of each of these things. And we don't have those. So to help us to get those, we have to use the ice table. So the ice is initial, change, and equilibrium. So I'm going to get rid of this for a minute so I have a little more space. So the initial concentration of Fe3 plus and SCN minus can be determined by using the volumes on the bottle. And I'm going to the volumes and the concentration on the bottle. And I'm going to do this for the first sample, which involved using 5 milliliters of the iron solution, 1 milliliter of the thiocyanate solution, and 4 milliliters of water for a total volume of 10 milliliters. So the concentration of Fe3 plus can be found by using the dilution equation. C1V1 equals C2V2, where the initial concentration is 2 times 10 to the minus 3 molar, because that's what you're given. That's what's on the bottle. The amount that we used of this in the first solution was 5 milliliters, and this is actually constant. We're going to use 5 milliliters in all those solutions. The final concentration, that's what we're trying to figure out, and the total volume for all of the solutions is 10 milliliters. When you do this math, you find that C2 is 1 times 10 to the minus 3 molar. So the initial concentration of iron 3 plus is 1 times 10 to the minus 3 molar. What about the initial concentration of SCN minus? Well, it turns out you could do this exactly the same way. C1V1 equals C2V2. I'm not going to write it again. But we're starting off with 2 times 10 to the minus 3 molar thiocyanate solution. But for the thiocyanate, for the first solution, we don't take 5 milliliters. We take 1 milliliter. The final concentration is the variable. That's what we're looking for. And the total volume, because it's in the same solution, is 10 milliliters. When you do this, you find that the concentration is 2 times 10 to the minus 4 molar. So we can plug that in. Now, the initial concentration, usually we don't put the units here. The initial concentration of FeSCN2 plus is going to be 0 because we don't have any of that. Now, how do we find the equilibrium concentration? Well, the equilibrium concentration of the FeSCN2 plus, if that's what exists, is going to be found by using spectroscopy. So Tim already talked about how we have to make a calibration curve. We then have to use that calibration curve to determine the concentration of FeSCN2 plus. To do that at equilibrium, we are going to use the calibration curve again, as I said, FeSCN2 plus. And the absorbance of this solution with 5 milliliters of Fe3 plus and 1 milliliter of SCN minus and 4 milliliters of water, when we measured the absorbance, it was 0.1202. And in the first part, we're going to use the calibration curve that we generated in order to determine the concentration of this absorbance. And the calibration curve had an equation, y equals 5660x plus 0.032. Well, absorbance was on our y-axis, so it's a y value, so we plug in 0.1202 equals 5660x plus 0.032. Subtract 0.032 from this, and then divide by 5660, and we find that x equals 1.558 times 10 to the minus 5 molar. So this is the change. We started with none of the FeSCN2+, and at equilibrium, as measured by spectroscopy, the concentration is an additional 1.558 times 10 to the minus 5. Well, if we form this amount of product, we must have lost this amount of iron and this amount of thiocyanate. Note that if we're investigating the other pathway, we have to lose double the amount of thiocyanate because there's two of them in the other pathway. But here we're going to subtract 1.558 times 10 to the minus 5, and here we're going to subtract 1.558 times 10 to the minus 5. And at equilibrium, so we simply take this and subtract this, we have 9.8 times 10 to the minus 4 as the equilibrium concentration of iron 3+. Then for SCN minus, we have 1.8 times 10 to the minus 4. And the equilibrium concentration of the iron thiocyanate is the same because it's 0 plus that number, so 1.558 times 10 to the minus 5. Now that we have all of these equilibrium concentrations, what we can do is use the equilibrium constant expression. So KEQ equals the concentration of products over reactants. The concentration of the product is 1.558 times 10 to the minus 5. And the concentration of the first reactant, iron 3+, is 9.8 times 10 to the minus 4. And the concentration of the other reactant, SCN minus, is 1.8 times 10 to the minus 4. And when you do that math, you find that the equilibrium constant is 86. So, in the past, 5 or 6 minutes, I've gone over how to find the equilibrium constant for pathway 1 for the first solution. Note that you're making 5 solutions and you're doing 2 pathways, which means you'd have to do this math 10 times. Nobody wants to do this math 10 times, right? It's a pain in the neck. So what we're going to do next is Tim is going to show you for the first pathway how we can use Excel to help us do this repetitive math in such a way that it's much more efficient. So essentially we only have to do it once and then it'll calculate it for us 10 times. Although there are a few differences, it won't exactly work that way. That's basically how it works.
So now I'm going to take some time to go over the math in Excel for this experiment. Now, this particular Excel sheet is actually going to be available to you in its entirety on Blackboard. However, when it's on Blackboard, these absorbances will not be filled in, and these absorbances will not be filled in. But the remainder of the sheet will be filled in as you see it here. This graph is based on these absorbances, so this graph will be blank when you come into lab, this graph being the calibration curve. When you come into lab, your TA is going to have 3 solutions made for you. Those 3 solutions will be 5 times 10 to the minus 5, 1 times 10 to the minus 4, and 1.5 times 10 to the minus 4 molar iron thiocyanate product solutions. Once you measure the absorbance of those 3 solutions and plug them in on this chart here, your graph will auto-populate and you'll get the line of best fit automatically for your data. So make sure you do that when you come into lab. So once you do that, there's a few things that you need to do. The first thing that we need to do is we're going to do the first pathway. So in order to figure out the first pathway for the sake of this video, I need to do some math. In this area, I've already put in the absorbances of my 5 solutions as Colin and I measured them when we performed the experiment. Your numbers will not be the same as these, which is okay. So just make sure you're using your numbers and not the numbers in the video in order to do your calculations. So the first thing that we need to calculate is the concentration of the iron thiocyanate product. So in order to do that, we're going to do exactly what Colin just did on paper, but we're going to do it in Excel. So in order to find the concentration of our product, we need to use our line of best fit. So if we rearrange this equation to solve for x, because we're going to use the absorbance y to solve for the concentration x, then we need to take our y value, we need to subtract 0.032, and then we need to divide by 5660, which is just solving this equation for x. When we do that, we find that we get a number very similar to the one Colin showed you before of 1.558 times 10 to the minus 5th molar. Now we need to do that for each of these other values. However, because it's Excel, we don't have to type that in every time. Since these two numbers, the plus 0.032, or minus in this case, and the divided by 5660 are typed in, the only number that'll change is the absorbance. So if the absorbance is the only number that changes, we can just drag this down, and it'll calculate our concentration for each of those absorbance values. So the next thing we need to do is we need to find our initial concentrations, because in order to complete that ice chart and find the equilibrium concentration, we have to find the initial concentration and then subtract out this changed concentration. So in order to find the initial concentration, again, we're going to do exactly like Colin did. We're going to take the milliliters of the initial solution, we're going to multiply it by the concentration of the initial solution, and we're going to divide it by the total volume, which is 10 milliliters. So we'll find, just like Colin said, that it is 1.0 times 10 to the minus 3 of the initial concentration of the iron. For the potassium thiocyanate, or just the thiocyanate ion that we care about, we're going to do the exact same thing. We're going to take the milliliters that we use, we're going to multiply it by the concentration, and we're going to divide it by the total volume, which is 10 milliliters, which, again, as Colin said before, is 2 times 10 to the minus 4th molar. So now we can just drag these down, and we'll get our values for each one. But wait, we have a bunch of errors. Why do we have those errors? Well, if we look at any of these individual things, you'll notice that the values are being selected from the wrong cells, which means we need to lock some of these cells in. And by some of them, I mean specifically this cell, because we want this cell to change for each one. In the case of the iron nitrate, of course, it's the same value, but when it comes to the potassium thiocyanate, that value will change. So we need to put our mouse cursor somewhere inside of the red box, the red value, because it's corresponding to the red box. We hit F4 and it'll put our little dollar signs in, which tell Excel not to change any of the values. We do the same thing for the thiocyanate, again the red box, so we want the red value. We hit F4, we hit enter. Now this time, if I drag those cells down, we don't get any errors, and the numbers make sense. At this point, you can do a quick sanity check and make sure that your iron concentration is the same in each solution, which it is, and it should be, because we're using 5 milliliters of the initial solution every time. So now we need to actually solve for our equilibrium constant. So in order to solve our equilibrium constant, we need to find each of our equilibrium concentrations. So the easiest one to do that for is the iron thiocyanate, because we've already calculated that concentration here. So we can just click on that cell, and then when we drag down, it'll take each of the values from each of these cells. You'll notice that it rounded a little bit, which is okay. Next, we need to find the concentration of our iron and our thiocyanate. Well, as Colin said before, in order to find the concentration, we're going to take our initial concentration and we're going to subtract out the concentration of the iron thiocyanate. This is because in order to make iron thiocyanate, we need to use up our iron. And however much iron we use is how much iron thiocyanate we make. So we don't need to do any calculations with that. We just take our initial concentration and subtract out the concentration of the iron thiocyanate. We hit enter and we have our number. We do the exact same thing for the thiocyanate concentration, except we're going to start, of course, with our initial concentration of thiocyanate and subtract out the concentration of our iron thiocyanate 2+. When we hit enter, we'll see that these two numbers have filled out, and we can drag those values down so that they calculate for each of these scenarios. You'll notice that the numbers in this case are not the same for the iron in every solution because, of course, the concentration of iron thiocyanate changes in each solution. Now that we have our equilibrium concentrations, we can actually calculate our KEQ. So to calculate our KEQ, we're going to do exactly what Colin did previously. We're going to take the concentration of the iron thiocyanate, we're going to divide it by the concentration of the iron at equilibrium times the concentration of the thiocyanate at equilibrium, close our parentheses, and hit enter. And we'll see that just like when he did it on paper, we find that our value is 86. In this case, Excel's written it as 8.6 times 10 to the first power, but that's the same number. So now, just like before, we can drag down our KEQ, and we'll have our KEQ value for each of our solutions. The last thing that we'll need to do is we'll need to calculate the RSD of these values. I'm going to leave that to you. By now you've done that enough. You should know how to do that. So you can calculate the RSD of this KEQ. Remember, the KEQ is a constant number for any given reaction, so we would expect that if pathway 1 is occurring, for this RSD to be low. And if pathway 2 is occurring, then the RSD for pathway 1 should be high, because that means that the equilibrium constant, as calculated by this, is changing a lot over the values. Keep in mind these are relative numbers. When I say low and high, I don't mean 1 for a low RSD in this case, but I do mean that one of these numbers, one of these RSDs, will be significantly lower than the other one, and whichever one is lower is, of course, the reaction that's occurring. So one thing I want to point out to you is that when you do perform this math in class, when you perform it for pathway 2, it's important to keep in mind that the SCN in pathway 2's equation is using 2 SCNs. So when you subtract out the concentration of the iron thiocyanate from the initial concentration, you need to make sure you're subtracting 2 times the iron thiocyanate, because you're going to need 2 thiocyanates for each of these products. And also keep in mind that means when you actually calculate the KEQ, you'll need to square the concentration of the thiocyanate in that answer. The last thing I want to talk to you about in this video is the hypothesis. At this point in the course, we are no longer providing help to you in the lab manual for writing your hypothesis, so I just want to give you some general pointers in order to help you kind of be steered in the right direction. So, of course, as always in your hypotheses in this course, the most important thing and the first thing that should be present in your hypothesis is a prediction of the results. So in this case, what do I mean by the results? Well, the whole purpose of this experiment is to figure out which of these pathways is occurring. So your prediction should be which of those pathways is occurring. And if you look this equation up online, you can find that information fairly easily. So you'll want to make sure you have a prediction for which of these two is occurring. The next thing you'll want to have in your hypothesis, which will likely be multiple sentences, is the scientific reasoning that you're going to use to prove your hypothesis, or disprove it if you are incorrect about which pathway is occurring. So when I say that, what I mean is you should write about how you're going to use a varied set of concentrations in order to get a variety of KEQs, and how you're going to use that information in order to help you determine which of those equations is taking place. So that's all we have for you for this video. We hope that these videos are helping and that your course is going smoothly.